guys, welcome back to Joanne's Learning Hub. These are my notes on IGCSE Biology Inheritance. So what is inheritance? Inheritance is the transmission of genetic information from one generation to the next. Chromosomes are thread-like structures of DNA that carry genetic information in a string of genes. DNA is a chemical from which genes and chromosomes are made. A gene is a short length of DNA that codes for a specific protein. It is found on a chromosome. Alleles are different versions of a particular gene. A haploid cell is a cell with a single set of chromosomes, whereas a diploid cell is a cell with two sets of chromosomes. Mitosis is nuclear division, which gives rise to genetically identical cells where the number of chromosomes are equal. So as we can see here, the first cell, which is the parent cell, in that the DNA is replicated, and then mitosis occurs in the second step, where each chromatid splits. One chromatid from each chromosome goes into each daughter cell. So we can see two daughter cells produced, and both the daughter cells are identical to its parent cell. Mitosis occurs in growth, repair, and asexual reproduction. Mitosis is important in stem cells. Stem cells are unspecialized cells. So depending on what instructions they are given, stem cells can divide by mitosis to produce identical daughter cells that can then become specialized for a specific function. Moving on to meiosis. Meiosis is reduction division, which halves the chromosome number from diploid to haploid, resulting in 4 genetically different cells. So in meiosis, the cells divide twice, so only one copy of each chromosome passes to each gamete. This produces 4 haploid daughter cells. So now we can see that the DNA is replicating, then the homologous chromosomes pair together, so it's maternal plus paternal. Then after that, sections of DNA are swapped between the maternal and paternal chromosome pairs. The first division in meiosis occurs where homologous chromosomes separate. The second division is when a chromatid of each kind goes into each daughter cell. So we can see each of those 4 daughter cells produced, and they are different, genetically different from its parent cell. Alleles are different versions of the same genes. We have 2 copies of each chromosome, so we have 2 copies of each gene, and therefore we have 2 alleles for one gene. So one allele is inherited from the mother, whereas one is inherited from the father. Genotype is the alleles an organism has. It is its genetic makeup. It controls each of the organism's characteristics. Phenotype is the observable features of an organism. Alleles are of 2 types, dominant and recessive. A dominant allele is an allele that is always expressed if present. A recessive allele is only expressed if there is absence of a dominant version of the allele in the organism's genotype. Homozygous is when the organism has 2 identical alleles for a particular gene, whereas heterozygous is when the organism has 2 different alleles for a particular gene. So homozygous dominant is the organism has 2 copies of the dominant allele, whereas homozygous recessive is that the organism has 2 copies of the recessive allele. The dominant allele is given a capital letter, whereas recessive is given the same letter but in lowercase. Monohybrid inheritance is the inheritance of characteristics which are controlled by a single gene. 2 identical homozygous individuals that breed together can be called pure breeding because they only pass one allele and therefore there will be only one phenotype. So the first example we can see here is a homozygous tall plant being bred with a homozygous short plant. So we are naming the homozygous tall plant as T, which is its genotype, and the homozygous short plant genotype is t. So when we breed these two together, we'll get 100% chance of the offspring becoming tall, but all the offspring will be heterozygous. In the second example, two heterozygous tall and short plants are bred together. So we'll get the phenotypic ratio 3:1. So there is 75% tall and 25% short. So out of this, there is one homozygous dominant tall plant, one homozygous recessive short plant, and then we have two heterozygous plants. The third example shows crossing a homozygous recessive and a heterozygous plant. So here we will get 50% tall and 50% short. A test cross can be used to determine the unknown genotype of an organism by analyzing the ratio of phenotypes in the offspring. So for the first generation, we will breed a homozygous dominant with a homozygous recessive, and in the second generation, we will breed the heterozygous with a homozygous recessive. So here we are again using tall, capital T, and short, small t. So we get all heterozygous. Then we breed heterozygous, which is capital T, small t, along with small t, which is short. So we'll get 50% tall and 50% short. Codominance is when both of the alleles in a heterozygous organism are expressed, and so both determine the organism's phenotype. One example of codominance is the inheritance of blood group. So for a phenotype of blood group A, we have the genotype either IA-IA or IA-IO. For phenotype blood group B, we have either IB-IB or IB-IO. Then here is the example of codominance. For phenotype AB blood group, the genotype will be IA-IB, and for phenotype O blood group, it will be genotype IO-IO. So I is the gene and the superscript A, B and O represents the alleles. So the IA genotype results in production of antigen A in the blood. IB genotype, it results in production of antigen B in the blood. IO, however, does not result in any antigens being produced in the blood. In humans, there are two chromosomes responsible for sex determination. These are the sex chromosomes, and they can be labeled as either X or Y. Females have two XX chromosomes, and the XX combination allows female characteristics to develop. Males have an X and a Y chromosome, so the Y chromosome is what causes the male characteristics. As only the father can pass a Y chromosome, he is responsible for determining the sex of a child. So there is a 50-50 chance of a child having either XX and being a girl or XY and being a boy. A sex-linked characteristic is a feature that is coded for by a gene located on a sex chromosome, making it more common in one sex than the other. So in most cases, there are only alleles on the X chromosome, as the Y chromosome is much smaller. Since males have only one X chromosome, they are more likely to show sex-linked recessive conditions. Since females have two copies of X chromosome, there are three possibilities: normal, carrier, or diseased. An example of a sex-linked disorder is red-green color blindness. It is caused by a faulty allele carried on the X chromosome. So women need two copies of this recessive allele to be colorblind, whereas men only need one copy. So that means color blindness is much rarer in women than in men. So chromosomes are made of DNA, and DNA codes for a protein manufacture. So in a DNA molecule, certain lengths will contain codes for specific proteins. A specific length of DNA, as we discussed in the previous page, a specific length of DNA which codes for a specific protein is called a gene. So the gene contains very specific sequences of bases that orders the cell to join certain amino acids in a certain sequence. So DNA is found in the cell nucleus. It cannot move out because it's really big. So the cell needs to get information from their DNA into the cytoplasm. It does this using a molecule called messenger RNA, mRNA. So like DNA, mRNA is also made up of bases. It is a copy of a gene. So mRNA is made in a nucleus and it copies the DNA and then it moves into the cytoplasm. Once it's in the cytoplasm, it passes through a ribosome and the ribosome assembles the amino acids into protein molecules. The sequence of bases in the mRNA molecule determines the specific order of amino acids in the protein. Like, share and subscribe to my channel for more videos.